3, Section 2, Cell Organelles. Eukaryotic cells share many similarities. Cells have an internal structure. The cytoskeleton has many functions. It's made up of a network of fibers extending throughout the cytoplasm. It provides mechanical support and maintains the cell's shape, much like your skeleton inside your body gives your body support and shape. It provides anchorage for many organelles and systolic enzymes. It's dynamic. It's dismantled in one part and reassembled in another. This helps change the shape of the cell when necessary. It also plays a major role in cell motility. Several organelles are involved in making and processing proteins. The nucleus, for example, stores the genetic information. The nucleus stores genetic information and lies right inside the endoplasmic reticulum. The endoplasmic reticulum, often called the ER, is made up of a membranous tubule with internal fluid-filled spaces. It is continuous with the nuclear envelope. The rough ER is dotted in ribosomes. The two types of endoplasmic reticulum, both will transport proteins that are made by the ribosomes. However, the rough ER is covered in those ribosomes. They're especially abundant in cells that secrete protein. Proteins synthesized on attached ribosomes and inserted into cisternal space and folded into its three-dimensional shape. They're also attached to secretory proteins. They are putting transport vesicles in and sending to the Golgi bodies. They're also membrane factories. They make phospholipids. As the endoplasmic reticulum grows, vesicles move membranes to other spaces. The smooth endoplasmic reticulum lacks ribosomes. It contains enzymes for many different metabolic processes. It synthesizes oils, steroids, and phospholipids. For example, your sex hormones and adrenal steroids. In your liver, it functions to break down toxins, nitrogen waste from cells, drugs, and alcohol. Frequent drug use can lead to increased smooth endoplasmic reticulum for increased breakdown, which is why tolerance increases and higher doses are needed for some, and why frequent and extreme drug use leads to liver damage. In the muscles, the smooth ER stores calcium ions and regulates muscle contraction. The ribosomes link amino acids to form proteins. This is the site of protein synthesis. Ribosomes themselves are not surrounded by a cell membrane or a plasma membrane. They are not membrane bound. They are made solely of rRNA. There are free ribosomes that are suspended in the cytosol, and they make cytosol proteins. Then there's bound ribosomes, like we just talked about on the rough ER, or they're sometimes located on the nuclear envelope. They make proteins for cell membranes or export. Ribosomes link those amino acids to form proteins, and then vesicles are the membrane-bound sacs that will hold those proteins and move them around the cell. Vesicles also serve as short-term storage for other materials inside the cell. The mitochondria is responsible for supplying energy to the cell. This is the site of cellular respiration. It's not a part of the endomembrane system. Membrane proteins made by free ribosomes and ribosomes inside the mitochondria. Semi-autonomous, meaning it grows and reproduces independently. It's mobile, it moves on skeleton tracks. It has a double membrane, which creates internal compartments. The smooth outer membrane and the inner membrane are separated by this intermembrane space. The folded inner membrane, or the cristae, increases the surface area for the chemical reactions to occur. The fluid-filled space, enclosed by the inner membrane, a matrix, contains DNA, ribosomes, enzymes for cellular respiration, and this is the site of the cellular respiration we're going to talk about in future chapters. Its job is to break down sugars, fats, and other fuels in the presence of oxygen to generate Cells with high energy needs, like muscle cells, have large numbers of mitochondria. Mitochondria is also unique because the DNA that's located inside of it is a maternal type DNA. It came from your mother. So when the egg was fertilized when you were created, there were several organelles that were supplied by the egg, and the mitochondria happens to be one of them. So that's how they can do the DNA testing, and they follow the maternal linkage. Vacuoles are fluid-filled sacs that hold materials. Again, they're not part of the endomembrane system. Vesicles and vacuoles, which are the larger versions, they have varied functions. In plants, there's a large central vacuole, and it's responsible for holding large quantities of water. There are food vacuoles that are formed by phagocytosis. They fuse with lysosomes. Contractile vacuoles and freshwater protists pump excess water out and maintain the salt-water balance. They stockpile proteins or inorganic ions. They dispose of metabolic byproducts. They can hold pigments, store defensive compounds to defend the plant against herbivores, or the large vacuole can reduce the area of cytosol so the surface area to volume ratio can increase. The water storage that takes place inside this large central vacuole is what makes plants turgid, or it keeps them from wilting. So when that central vacuole is empty, the plant wilts. Lysosomes contain enzymes that digest material. They're found in animal cells. These membrane-bound sacs of digestive enzymes can hydrolyze food. They can consume whole cells, damage cell parts, and these enzymes are made by the ribosomes on the rough ER and modified in the Golgi. So it's all in-house. This is an example of compartmentalization. Enzymes work best at the pH of 5. Hydrogen ions are pumped from the cytosol into the lysosome. If a lysosome ruptures, enzymes are not very active in the cytosol, which is neutral, and this is what prevents accidental self-digestion. Massive rupture of many lysosomes can cause a cell to destroy itself by self-digestion. Centrioles are tubes found in the centrosomes. They're seen only in dividing animal cells. They're made up of microtubules in patterns of 9 triplets. They're found inside the centrosome, and they replicate and move to poles during cell division. The centrioles form cilia and flagella. These extend from the cell surface. They're surrounded by a plasma membrane sheath. They're anchored in the cell by a basal body. They're flexible protein wheels that connect microtubule doublets, and the motor proteins, these dynein arms, connect outer doublets and allow walking of dynein arms along microtubules. This causes bending and movement. While it requires ATP, it's how things move inside of our cell. So it's pretty cool looking. It almost looks like an alien, but this is how things move around inside of your cells. The ATP consumption allows that motor protein to work its way along the cytoskeleton itself. Plant cells have cell walls and chloroplasts. Here we can see each one of those little bubbles is a cell membrane surrounded by a cell wall, much like Robert Hooke saw when he first looked at the cork cells. These guys are living, and you can see the chloroplast inside of them. That's what gives it that green hue. Cell walls provide rigid support. They're also found in prokaryotes, some fungi, and some protists. Their job is to protect, support, and maintain shape. The thickness and the chemical composition differs from species to species among different cell types. Microfibules of cellulose embedded in a matrix of proteins and other polysaccharides provides the structural support that plant cells need. Chloroplasts convert solar energy to chemical energy. The plastids that are found in leaves and green organs of plants and algae, membrane proteins made by free ribosomes and ribosomes inside the chloroplast. This site of photosynthesis takes the energy from the sun, goes through a process of chemical reactions, and creates chemical energy in the form of glucose. chloroplast itself is a double membrane, creating central internal compartments. There's an intermembrane space called the stroma. It also contains some DNA. And then inside there are stacks, and it's a little hard to see here, but these are stacks of thylakoids. Those thylakoids just look like flattened coins, and those thylakoid membranes are where the chemical reactions of photosynthesis are actually taking place. Some other plastids would be amyloplasts, which are colorless plastids that store starch in roots and tubers, like potatoes. And chromoplasts. They store colored pigments for fruits and